to the sensory nerves of the external body wall. We also knew a long time ago that visceral pain was associated with greater autonomic and emotional responses than other types of pain. And importantly, Mackenzie wrote in 1906, the viscera are insensitive to those stimuli which produce pain when applied to the external body wall. The visceral tissues can be cut, torn, and burnt, and the individual remains unconscious of any sensation. So Mackenzie was well ahead of his time in summarizing things about visceral pain, and explains here that all of the usual stimuli that produce pain when applied to the skin or to other tissues in the body don't produce pain when applied to the viscera. And so we can look at a simple cartoon here of pain associated with visceral structures and associated with the skin, and ask several questions. We know that visceral pain is referred. We know that it's poorly localized, but is injury required? As Mackenzie said, it's not. And so you have to ask yourself, why not, when we know a lot about cutaneous pain? And Mackenzie wrote, the pain can be reduced by visceral stimulation, is easily demonstrated if one employs an adequate stimulus. And so an adequate stimulus goes back to language that was advanced by Sherrington, and it relates to the fact that the adequate stimulus to produce pain differs in different tissues. It wasn't appreciated until Mackenzie advanced some work that he had been doing in the late 1800s and early 1900s. And so the adequate stimulus for skin, we know, is damage or threat of damage to tissue. Sherrington provided the terminology for us of what refers to a nocuous or noxious stimulus, what is a nociceptor. But we know that in the viscera, the adequate stimuli are stretch or distension, traction or pulling on the mesentery, inflammation of the tissue, ischemia, things of that nature. We know that cutaneous pain is not referred, localization is excellent, hypersensitivity develops when skin is injured. In the viscera, we know that pain is referred, localization is poor, but hypersensitivity also develops in the viscus and also in the skin and deep tissues associated with that particular visceral organ. And again, visceral pain is not felt at its source. So a couple of important distinctions going forward. Nociception, this was a term that was introduced by Sherrington to make clear the distinction between detection of a noxious event and the psychological and other biological responses to it. And it's defined by the International Association for the Study of Pain as the neural process of encoding noxious stimuli. Pain, in contrast, is defined as an unpleasant sensory and emotional experience associated with or resembling that associated with actual or potential tissue damage. And pain is always subjective. And so when we study mechanisms of visceral pain in non-human animals, we're studying nociceptive processes. And pain and nociception go hand in hand most of the time, but nociception does not equate with pain necessarily. So what's a nociceptor? Sherrington provided the baseline information for us. Nociceptors are receptive endings in skin. He was working with skin and subcutaneous tissues when he was squeezing and cutting and doing other things to skin. And he defined noxious stimuli as adequate when they excited nociceptors by threatening damage to skin. And we know that the adequate stimuli differ. For skin, it's cutting, crushing, burning, chemical stimuli, viscera, traction on the mesentery, distension of hollow organs, inflammation, ischemia. And for muscle, joints, and bone, it's associated with chemical stimuli released from nerve endings or from tissue in those, or from those tissues. And here's a cartoon just to emphasize what Sherrington was talking about. Here we have a piece of skin with a receptive ending in the skin, its sensory axon conveying information into the spinal cord, where traditionally it crosses to the contralateral side and then ascends to supraspinal sites. The cell bodies of these neurons are in the dorsal ganglion. Technically, this is what a nociceptor is. It's in the skin and it's the receptive ending. What has happened over time, however, is that not only is it this sensory receptive ending, it is the axon associated with it and the cell body associated with this receptive ending that is considered to be a nociceptor. And so studies occur with respect to studying out here in the periphery, studying these axons, and studying these cell bodies, all which are part of a nociceptor. Why are they important? Just to emphasize that they are the functional unit of the nociceptive pain sensory system. All information that is available to the central nervous system about a noxious stimulus is encoded by nociceptors. They contribute selectively to different forms of pain. There's mechanical pain, thermal pain, itch, and from the viscera, there are other types of sensations associated with activation of nociceptors. Importantly, when the activity of nociceptors is blocked, pain resolves for the duration of the local anesthetic action. And you know this if you've ever gone to a dentist and had a local anesthetic injection before some process or some procedure is done in the mouth. But we know this also from human neuropathic pain situations. It goes back to the work of Gracely and his colleagues in the early 1990s. Also with respect to the viscera, both the bowel for irritable bowel syndrome and interstitial cystitis in the bladder. Don Price and his colleagues showed that you can locally anesthetize the rectum and the distal colon and then distend, and the response is significantly reduced. And in patients with IBS, their complaint of pain is significantly reduced as well. So blocking the input or the activity in nociceptors can have a significant influence on the sensation of pain in humans. And then the question arises, are there visceral nociceptors? And it gets a little bit complicated because in the 1920s, early 1920s, Langley, who anatomically identified sensory fibers from the viscera coursing with or running with the motor axons of the efferent sympathetic division of the autonomic nervous system. And he named them afferent sympathetic fibers, which others shortened to sympathetic afferents. And that turns out to be an unfortunate oxymoron. As a pharmacologist by training, we knew that the sympathetic and the parasympathetic divisions of the autonomic nervous system were basically motor neurons that produced piloerection and vasoconstriction and vasodilation. And so to call these fibers, these sensory fibers from the viscera, sympathetic afferents was confusing. And if you read the literature from the 1930s up until even in the 1970s and 1980s, getting started in the field of visceral pain is confusing because of some of this terminology. So we know that the viscera are innervated by sensory neurons, but are there any of those neurons which are nociceptors? And at that time, the thinking was nociceptors have high thresholds for activation. And in 1982, Fernando Severo published a paper looking at the axons innervating the gallbladder in the ferret. And so he looked at biliary pressure, shown up here across the top of these three different neurons that he illustrates, and their response as he increased the pressure inside the gallbladder. And I just highlighted here for you the fact that there is an intensity-dependent increase in response in this particular neuron and in the others, and at high pressure, there's a increased response here. And so we have to assume that distending pressures in the gallbladder of 30 or 40 or 80 millimeters mercury are painful. There's no behavioral evidence that this is so, but it makes logical sense and is, in fact, has been subsequently documented that high intensity distension of bladder, colon, gallbladder, esophagus, stomach are associated with activation of high threshold mechanosensitive afferents that innervate those organs. Now, the IASP's current definition, which was recently changed about a nociceptor, is as follows. It is a high threshold sensory receptor of the peripheral somatosensory nervous system that is capable of transducing and encoding noxious stimuli. Now, that's true for most cutaneous afferents, but not uniformly the case. And as I'll show you, when you examine and understand the properties of nociceptors, threshold for activation is not a property that is required a receptive ending to be classified as a nociceptor or to function as a nociceptor. So the existing belief about threshold was understood to be limiting even in the 1960s, when Ed Pearl had shown that nociceptors have special properties or different properties from other neurons. And those properties are quite simple. They encode noxious inputs to the central nervous system, which we know, but they sensitize after tissue insult. And this is a property unique in the sensory innervations of bone, joints, muscle, and skin. Other somatosensory receptors do not sensitize. And what's sensitization? The response magnitude increases to a given intensity of stimulation relative to control circumstances. Response threshold decreases, and spontaneous activity may also increase. Nociceptors also have an efferent function. That is, they're associated with the release of peptides and other mediators from their sensory endings in tissue, a phenomenon referred to as neurogenic inflammation, which has been known for a long time. And some of them, as I'll show you, are mechanically insensitive. So let's get to the clinical circumstance. Is there sensitization that is obvious or apparent in these visceral pain states? This is work from James Ritchie, published in 1973, in which he looked at patients and asked them to report pain as he distended the distal colon in these individuals. And you can see here's the response in normal subjects. And when they were distended with a volume of air of about 60 milliliters, a threshold for producing a response, less than 10% reported that as painful. In the IBS patients, that same distending volume produced a pain sensation in more than 50% of those patients. So this is characteristic of sensitization. The response threshold has been reduced. Here it's about 60 milliliters of air, and here it's maybe 40 milliliters of air, and response threshold is much greater. So there's this leftward shift in this psychophysical or stimulus response function. Same is true in the urinary bladder. This is work from Tim Ness. He looked at interstitial cystitis subjects and healthy subjects. He asked them about the unpleasantness of bladder filling and the intensity of the sensation. And he found that in the IC or interstitial cystitis subjects, there was this leftward shift in both their unpleasantness and intensity. Again, visceral hypersensitivity shown by a change and a leftward shift in their psychophysical or stimulus response functions. The other thing that happens in visceral sensitization is that the area of referred sensation is significantly expanded. And here's work from Swarbrick and Dawson in the 1980s. They looked at normal patients and IBS patients and distended their colon, the ascending colon, the transverse colon, and the descending colon. And what happens is that in each of those circumstances, the area of referred sensation, shown here in yellow, increases significantly in size. And this is a reflection of what we now call central sensitization. Central sensitization was made popular by work by Clifford Wolff in the early 1980s. it became all the rage and became the explanation for all types of pain states. But I take you back to James Mackenzie in 1909, and he wrote that if the visceral, and I inserted visceral because he was talking about visceral organs, if the visceral stimulus be of sufficient strength, it may leave an irritable focus in the spinal cord, as shown by a persistent hypersensitivity of skin and muscle. So that's illustrated over here on the right. Here is the input from a skin nerve into the spinal curve, and it's of sufficient strength to cause this, what he termed irritable focus, which we would now call central sensitization, and excited a muscle nerve that illustrated or generated sensitivity or hypersensitivity in the muscle. And this became what Mackenzie called a visceromotor response. And it's because when he excited the viscera, there was also an increase in muscle hypersensitivity, illustrated by a contraction of the muscle. And that is how one can understand or test visceral pain in non-human animals, by distending a hollow organ and measuring the contraction of the muscle response that's produced. The other thing is that he was ahead of his time, James Mackenzie, in describing cross-organ sensitization. Here is illustrated input from one viscus that produces this irritable focus that generates a sensitization in another organ. And it turns out that cross-organ sensitization is today appreciated as a significant clinical problem. The people I'm working with at present at the University of Chicago are a group of gynecologists, and we've been studying women with dysmenorrhea. And it turns out that if you carefully examine and interrogate these women, about 60% of them have symptoms of irritable bowel syndrome, according to the Rome 3 criteria, and about 40% of them have bladder hypersensitivity, which you can establish by a simple drinking test, having them drink a lot of fluid and asking them when it becomes, when the first urge to urinate is produced and when the first unpleasant sensation is produced. They all have bladder hypersensitivity and colon hypersensitivity, although their main complaint is dysmenorrhea. So cross-organ sensitization has become a significant clinical problem. So you might ask, and this is a bit of a digression for those of you who don't know how you could study visceral nociceptors. And what I've shown over here is a piece of the colon from a mouse or a rat, and I've illustrated where there are receptive endings in the muscle, near the mucosa, somewhere between the muscle and mucosa, in the serosa. And this is illustrated here as the pelvic nerve, but this would be true for any organ and the nerves that innervate it. Well, what you do is you take this piece of tissue, you slice it longitudinally, and you pin it in a dish, and you infuse it with a Krebs solution that's oxygenated. And what you can do then is you can probe the surface of this colon. This is the mucosal surface up. And you can probe until you find a receptive ending that responds. You can record from these nerve fibers over here in the recording chamber. You can also then stretch the tissue. You can stroke the tissue with a feather, if you were, or a small piece of tubing. You can isolate the receptive ending with a brass ring or a brass tube, and then pour in hot solution or cold solution, or you can pour in a chemical solution. And this is what it looks like in the dish. This is from Ben Feng's work. And illustrated by the red broken line is the actual piece of colon that is in the dish. This is a rake of hooks that are connected to the antimesenteric border that's connected to a mechanism that we can stretch the colon in a controlled way. This is a stimulating electrode that allows one to move up and down the colon and across the width of the colon to produce an electrical stimulus. These circles represent the spread of current. And so you can rapidly and effectively search for receptive endings in a non-biased way, because the electrical stimulus will activate a chemosensitive ending, a mechanosensitive ending, and a mechanically insensitive ending. And that's what was done in a number of studies, and I'll illustrate some of the results of that in just a minute. And you can also stretch this tissue. So you can do a lot with a preparation like this to study the innervation of the bladder, the colon, the stomach, any tissue that you can put in a dish like this and is big enough to study. And when you do that, this is what happens. You find that there are a number of afferent endings that have different properties. And we focused mostly on mechanosensitive endings, but we used electrical stimulation to find the receptive ending in the tissue. And so the classes of these afferents are muscular mucosal, serosal, and mechanically insensitive. And these red dots represent the stimulus artifact from the electrical stimulus on top of the colon. And these spikes that follow at different delays are action potentials of single fibers that are recorded, in this case, in the pelvic nerve. And you see the latencies are different because this is probably in a delta fiber, and these are C fibers that have slower conduction velocities. So you can tell something about the fiber you're recording from by the latency to its response to an electrical stimulus. And then what we do is we test that receptive ending for probing, and all of these mechanosensitive endings respond to blunt probing of the receptive ending. But not all of them respond to stroking of the mucosa or stretch of the organ, as I showed you before. So this is a ramped stimulus that goes up to maybe 40 or 45 millimeters mercury, and it gives you a nice robust response. And when you relax the stimulus, the responding stops. And so that's a muscular mucosal ending because it responds to stretch and stroking. This responds only to stretch, so it's a muscular ending. This responds only to stroking, so it's a mucosal ending. This doesn't respond to either stretch or stroking, but it responds to blunt probing. And so it's been designated a serosal ending, but that's likely a misnomer, and what it actually is not clear. And at the bottom here, we have an ending that responds to electrical stimulation, as you can clearly see, but doesn't respond to any mechanical stimulus. So this is a mechanically insensitive afferent. And when you do these experiments, you find that some of these mechanically sensitive endings have low thresholds for response, and some have high threshold for response. And so as we talked about before, the high threshold responders you would assume would be those that are nociceptors. And so this is work that was done in the 90s. The response to distension is plotted vertically. This is the distension, in this case, of the colon. And you see there's a group of fibers, about 75% of the population, that have low thresholds for response, somewhere between 5 and 10 millimeters mercury distension, and they continue to encode the intensity of stimulation into the noxious range. And so you have to ask yourself, what is a low threshold fiber in the colon doing encoding the intensity of stimulation into the noxious range? Here's the high threshold group of fibers. They don't begin to respond until about 30 millimeters mercury, and they then faithfully encode the intensity of the noxious stimulus. But you're getting even a bigger response from the low threshold fibers. So that's a little curious about the visceral innervation. And then when you go and you look at the literature or you do the experiments yourself, you see that this is true for every hollow organ you can study: gallbladder, esophagus, stomach, urinary bladder, colon, uterus, even the ureter. The other thing is that you find out that both low threshold and high threshold fibers sensitize. And coming along here is going to be the response to stretching the colon of a low threshold and a high threshold fiber. You can see the threshold when you extrapolate it down here is very low for this example, and for the high threshold fiber is about 25, 30 millimeters mercury, something like that. What you can do, because we've isolated these receptive endings with this with a brass tube or a brass square, is we can apply a chemical stimulus to it, an inflammatory soup that contains serotonin, protons, bradykinin, prostaglandin, things that you want to put into it, and you apply that to the receptive ending, not to the entire colon, just to the ending, and you wait about 3 minutes or 5 minutes, and then you distend or stretch the colon again, and what you see is that the threshold for the high threshold fiber has now dropped down to be somewhere near what this low threshold fiber is. This fiber probably has some spontaneous activity, but also has a lower threshold response. You can see the response here is greater after desensitization than it was before, and that's true here with the high threshold fiber as well. And then you can wash out the stimulus and remove the ring, and you can see that responses return back to a pre-sensitized state. So both low threshold and high threshold fibers sensitize. They also both encode into the noxious range. And so my interpretation of that is that both low threshold and high threshold fibers that innervate the hollow organs of the body have the ability to contribute to pain because they sensitize and they encode in the noxious range. So there's a couple of contemporary things that are topical and a reminder. The reminder is that the visceral innervation is unique, and I'll show you that, illustrate that in just a minute. And some nociceptors are mechanically insensitive. These are termed silent or sleeping nociceptors. This is work that was done largely by Deng Feng in the laboratory, and I'll show you an example of that. And then cross-organ sensitization was investigated by Pablo in an early study between colon and bladder in 2009, and then he wrote a review article that was also provocative that appeared in Autonomic Neuroscience in 2010 about cross-organ sensitization. So here's a cartoon of the gastrointestinal tract, also showing the uterus and the prostate. This is an unusual individual, doesn't have any thoracic organs, and has both a uterus and a prostate. But if you look at the innervation of this tissue, and you look at the bladder and the distal colon via the pelvic plexus and the pelvic nerve, this goes to the sacral spinal cord, S2 and S3 in humans. But if you look also at the innervation of the bladder and the colon via the lumbar sympathetic nerve through the inferior mesenteric ganglion, it terminates in the thoracic spinal cord. And so the lumbar and the pelvic nerve innervate the same tissues in these paravertebral ganglion. The largest nerve in the body, the afferent nerve, is the vagus nerve, and it innervates all of these tissues as well. And so you see the key message here, without knowing the details of this, is that each organ is innervated by two nerves, and the functions of each are different. And I'll show you that briefly here. Here's lumbar sympathetic nerve. That tissue I've shown you before. This is a piece of the colon. These are the different kinds of receptive endings. You don't need to worry too much about that, but just note that most of the endings in the lumbar sympathetic nerve that innervate the colon are out here on the mesenteric attachment. And so you can stretch and pull that, and they would respond. There are none along the anti-mesenteric border. In contrast, if you look at the pelvic innervation, pelvic nerve innervation, you see that most of these endings are not associated with the mesenteric attachment, but are associated with the distal part of the colon. And that few of these respond to stretch or distension, and obvious noxious stimulus in the colon, and about a third of these do respond to this noxious stimulus. So the innervation is important to nerves, different functions. And let's talk just briefly about these mechanically insensitive afferents, these silent or sleeping nociceptors. So some of them are also activated chemoselectively, and this was in a subsequent report by Bin that appeared in 2016. But the mechanically insensitive fibers look like this. Here again, we're stretching the colon in a dish, and there's a muscular receptor ending that responds. This mechanically insensitive fiber is not responding to any mechanical stimulus. Here you see it does not respond to probing of its receptive field, but we did the same thing. We applied an inflammatory soup over the electrically activated receptive ending that was mechanically insensitive. And after we did that, you see it begins to respond to probing. So it turns out that these mechanically insensitive fibers, if you look at a pie chart of the different types of mechanically sensitive and insensitive endings in the mouse colon, about 25% are mechanically insensitive. So they may be thermoselective, they may be chemoselective, but they're mechanically insensitive. And the key important feature of mechanically insensitive endings is that they acquire mechanosensitivity after sensitization or tissue insult or injury. And this proportion, 25%, is consistent with the number of cutaneous mechanically insensitive fibers that are found in human skin by studies of microneurography by Hermann Hanwerker and his colleagues in Würzburg and in Heidelberg. So it's a real thing, and they contribute to pain after tissue is insulted and injured because they acquire mechanosensitivity and provide new input into the nervous system, central nervous system. The final thing to talk about is cross-organ sensitization. Now, this was all the rage in the 60s and 70s because some investigators had reported these dichotomizing afferent axons, which meant that there was one cell body out here in the dorsal ganglion that innervated two tissues. And it turns out that that wasn't documented very extensively, but subsequently has become more interesting and more important. And so what we show here, or what I show you here, is the urinary bladder and the prostate. And what we can do is inject into the muscle of the urinary bladder the retrograde tracer that is taken up by these nerve endings and transported back here to this cell body. And in the prostate, we can inject a different dye. In this case, a blue dye that's picked up and transported back to the dorsal root ganglion. And so when you take out the ganglia about 7 to 10 days later, and you section the ganglia and you examine it under fluorescent microscopy, you can see that the blue dye that was injected into the prostate is found in a number of neurons in the dorsal ganglion. The ganglia is sort of outlined here in an oval shape. I haven't outlined it for you, but the blue dots illustrate the cell bodies there. And you see that they're also, when you switch filters and you look at a different wavelength of illumination, you can see that there are cell bodies that are illustrated from the dye that was picked up in the bladder. But then you look here and you see there are some that contain both the blue dye and both the red dye. And so this turns out to be, in these experiments, about 17% of the neurons in the dorsal root ganglia. This is in the sixth lumbar, first sacral ganglia of the mouse that innervate two organs. Could innervate three organs for all we know, or could innervate an organ in muscle or an organ in skin. We just don't know because those experiments have not been done. But nevertheless, the fact that cross-organ sensitization has become a significant clinical problem, there is now an alternative explanation to the irritable focus in the spinal cord of the central sensitization that could contribute to the sensitization of an adjacent organ. And to show that this occurs, we looked at inflation or distension of the urinary bladder in animals that had had their prostates inflamed. And so this is the control response here in black to distension of the urinary bladder. And this is the responses 7, 14, and 21 days after inflammation of the prostate. And this is at a time when the inflammation is waning, of course. It doesn't last for 21 days. By 21 days, the evidence histologically of inflammation is largely gone. But you see here that the response to bladder distension is highly exaggerated here. And so this is organ sensitization, cross-organ sensitization, because the prostate is inflamed. The bladder has no inflammation by any histological measure that we made, yet it is sensitive to distension. If you look at the recordings from control-treated and inflammation-treated animals, and you record from bladder afferents, these are not prostate afferents, these are bladder afferents, you see, excuse me, you see a typical control response, threshold here, about 20 millimeters mercury, increasing responses as you increase the intensity of distension across the horizontal axis. And if you look at the zymosan-treated animals, look at the increased response magnitude here and even greater here. And you see that there's an after discharge that persists long after the termination of the stimulus. So the bladder afferents are sensitized, and the responses to bladder distension are also sensitized after the prostate is inflamed. So we don't know everything about viscera. We know more about visceral nociceptors and sensitization and things of that nature than we did many years ago, but there's still many things we don't know. We have no idea what molecules in tissue initiate and sustain afferent neuron or organ sensitization. And people have looked at numerous things, a variety of immunomodulatory molecules, growth factors, mast cell clusters, individual amines, monoamines, and there is no single molecule that has been shown to be responsible for the sensitization of either the afferent fibers or the organ. You can certainly sensitize an afferent fiber with serotonin alone, for example, but when you use a serotonin receptor antagonist to block the effects of serotonin in an animal, the sensitization of the organ remains. So it's highly likely that other molecules contribute to the process of sensitization and sustain it in the presence of a variety of pharmacological manipulations to block the actions of immunomodulatory molecules or growth factors. You can show things in mice and rats that just don't work out in human circumstances with respect to irritable bowel syndrome, interstitial cystitis, any of the other visceral disorders. We also don't know yet, and it's intriguing to me that we don't know about this because people have studied the enteric or the intrinsic nervous system for decades, even before we began to study visceral afferents. And so we don't know what the interaction is between these efferent innervations of the autonomic nervous system or the intrinsic enteric nervous systems of the gut and the afferent systems that are associated with sensory processes. So this is the interactions between the intrinsic primary afferents and the extrinsic primary afferents. They exist, they must certainly exist. When Pablo was in the laboratory looking at Vesicular glutamate transporters, there was obvious evidence histologically that these neurons contained vesicular glutamate transporters and were in close association with the sensory afferent terminals, but the interaction didn't appear to be synaptic and it didn't appear to be physical. But there must be interactions because these things occur quite frequently. And then I didn't talk today about tonic descending effects, either facilitatory effects or inhibitory effects on visceral pain states. Experimentally, it's been documented, and for a while, all the rage in the clinical literature with respect to irritable bowel syndrome, at least, was conditioned pain modulation. And they did these experiments by using cold water immersion of the limb and measured the response of people with irritable bowel syndrome and the response of controls. And they found out that the people with irritable bowel syndrome had less inhibitory modulation. Those studies have been very, very difficult to reproduce, and it's highly likely that it's dependent on the conditioning stimulus that's employed. And so the extent to which tonic effects on spinal processing are important in visceral pain states is unknown, but it's clear that there are likely significant influences from supraspinal sites that we have yet to ferret out. What don't we know and what's new on the horizon? Both of these things are important, I think, because they can be affected by peripheral mechanisms. I'm a pharmacologist. Dave's a pharmacologist. Others of you probably know a lot about pharmacology. I'm not a big fan of taking drugs systemically to attack what might be local problems that could be controlled by other mechanisms. And so neuromodulation is one such way to produce a localized effect by electrical stimulation of peripheral nerves. Transcutaneous electrical nerve stimulation has been around for a long time, but people are now stimulating spinal nerves, dorsal root ganglia. I know Ben has been working on this. And the challenges are many, but the reward could be significant because this can be stimulation that's localized to a spinal nerve or a single dorsal root ganglia. There are challenges that include the intensity of stimulation that is effective, the frequency of stimulation. One might think that you could use frequencies that are low, like 2 or 10 hertz, or high that are 100 hertz, but people are looking at stimulation intensities of 1,000 hertz and higher. And it's hard to understand how that has any influence on the mechanisms that are associated with neural transmission. Access to tissue is going to be a problem if you have to implant these electrodes. But I think neuromodulation has a future, and if they resolve some of these issues, it could be useful. More likely to be useful sooner is epithelial signaling. The bladder urothelium and the gastrointestinal epithelium have been targets. Colleagues of mine at the University of Pittsburgh have published studies on optogenetic excitation of colon epithelium that activates mechanosensitive colon afferents. So here's a cartoon taken from Sarah Najjar's Trends in Neuroscience publication earlier this year. And what you see here is a part of the colon with the colon epithelium here highlighted. And there are a variety of different cell types that are a part of the epithelium. There are I cells and L cells, enterocytes, tuft cells, enterochromaffin cells, and what they show here is nerve endings beneath these cells here along the interior part of the cell. And you see that there are P2X receptors, there are vesicular glutamate transporters releasing or controlling glutamate release, AMPA receptors, NMDA receptors, mGlu-R receptors. Here you've got acetylcholine associated with release affecting nicotinic receptors. here in the tissue. Serotonin is released from enterochromaffin cells. The gut, in fact, is the largest reservoir of serotonin in the body. Everybody has targeted serotonin receptors to control visceral pain, particularly in the colon, and the effects are modest. Anyway, that's what these people are doing. And here's evidence from a study that appeared in 2018. They used the preparation that I showed you earlier, where you can look at mucosal, muscular mucosal, and serosal mechanosensitive endings. And here they've done the same thing that I showed you before. They've probed the tissue, they've stretched the tissue, and they've brushed the tissue, and they found all 4 different types of mechanosensitive endings in the colon. And what they had done is that they had previously treated these mice to insert into different numbers or different cell types in the epithelium laser or blue light activated channels that could activate the cells to release whatever they released. And so what you see here is blue light stimulation of a mucosal, a muscular mucosa, and a serosal fiber. And these are mechanosensitive fibers now being activated by activation of a cell in the epithelium. So there's clearly evidence here that something is being released when this cell is being activated that can produce a response in a mechanosensitive cell. So this is kind of exciting because it suggests that one might be able to develop a pharmacologic probe that would activate a selective subset of cells in the epithelium or block the activation of those cells and prevent a mechanosensitive response. So that concludes what I wanted to talk about today. If you have the energy and the time and you want to walk up a trail in the mountains, you're often rewarded with beautiful flowers in meadows like this before you get to the lake and have your lunch. So I'll end things there and be happy to answer any questions that you might have. And I thank you for seeing me today and listening as well. Thank you.
Thank you very much, Jerry. Much appreciated the time and effort that you put in preparing this lecture, which I must say that with Carly here, we were, we both worked with similar setups as you have shown, at least some of them. And it brought the good memories of spending 8, 9, 10 hours in the lab trying to get signals from a fiber, from one. Those are difficult experiments. I open here the space for questions from whoever would like to make some. Marcelo, go ahead.
Thank you so much, Jerry. It's been a wonderful talk, and especially for many of us who are not working in the field. So we got a very nice overview, I would say, and you gave us a good historical perspective on how things have been developing and how knowledge has increased over the years about visceral sensations. So it's been wonderful.
Well, thank you, Marcelo.
I wonder, how do you relate these receptors to other type of sensations, like, for example, hunger or satiety? I mean, it's not exactly pain, but these types of sensations may become painful at some point. How is this related?
Well, it's a good question, and it's interesting. What I did not emphasize at the outset is that the receptive endings in the viscera are active most of the time. So, for example, you have receptive endings that produce sensations of which and produce effects of which you are unaware. You're typically unaware of changes in your blood pressure or changes in your heart rate unless it's some significant change. But those endings in the vasculature and in the viscera are always active. And when you eat those receptive endings generate responses to release things in the organ to help you digest your food and increase motility. So there's lots going on in the viscera which we don't sense most of the time. It's only when we have the sensation of hunger. And I don't deny that hunger is a real sensation that affects a significant population of undernourished people in the world. But when I get hungry, I'm probably not really hungry. I'm probably just interested in having a little piece of chocolate or something. So those sensations span a huge range from the trivial, my piece of chocolate, to maybe the life-threatening in individuals who are undernourished all of the time. And so I don't have a good answer for you, Marcello, how those sensations arise. They're perceived and interpreted in the central nervous system in the brain, likely having associated with some learning experiences, but also with real life-threatening experiences in the absence of nutrition.
So you wouldn't, I mean, or maybe this is it's not a good question. I mean, so there is no relation, for example, to hormones like ghrelin, or those are like parallel systems that have nothing to do with each other.
No, I'm sure that ghrelin and all of these other hormones that are related with changes in motility and changes in absorption and secretion in the gut and in other organs play a huge role and probably contribute to the, when ghrelin is there, it probably contributes to the sensation of satiety or hunger or some other thing. I haven't studied those. The study of actual nutrients and amino acids and fats and things on sensory endings in the gut, particularly in the stomach, precede by decades the studies that are associated with pain sensation. And so if you go back in some of the earlier literatures, French investigators were using sharp electrodes to penetrate dorsal root ganglia neurons, and then looking at responses to fats and to acids and to other proteins instilled into the stomach. And that was going on in the 40s and the 50s, as soon as they had amplifiers that were good enough to amplify a signal for them.
Okay. Then you have referred mainly to the bladder and to the GI, to the gastrointestinal system. What about other organs? Like, for example, those that don't have a light. I mean, like, for example, the thyroid gland or the liver or the pancreas, for example. Those are organs that don't have any nociceptors, or they do have them.
Well, that's an interesting question. I think the general thinking is that parenchymatous organs like thyroid and liver are not innervated with sensory neurons except for the vasculature within them and for the capsule that might contain the organ. So that's not to say that there's not excruciating pain associated with pancreatitis or pancreatic cancer or liver cancer, but it's generally been assumed, despite people trying to study this, that the parenchymatous tissue is not innervated as are muscle layers and other parts of the organ. There was a study done in Ashley Blackshaw's laboratory a while ago looking at the pancreas in an in vitro preparation. I don't know if you've ever tried to take the pancreas out of a rat or a mouse and put it in a dish, but it's like scrambled eggs. And so it's very difficult to actually apply a stimulus. And so those experiments are not the lowest hanging fruit. And so I don't deny that there may be innervation in those organs, but the general thinking is not in the parenchymatous organs.
Okay. And I have one last question. It's referred to the central nervous system, but why has it been so elusive to find a cortical area for visceral sensation? I mean, because, you know, we have the homunculus, we know exactly what happens in the somatic system, But with the visceral system and for conscious sensations, it has been not so clear, at least.
Yeah, well, Penfield's homunculus doesn't show the viscera very well on the cortical surface. It's interesting. There have been some studies done, one by a graduate student in my laboratory many years ago, and one by Vanya Apkarian, also many years ago, recording in the thalamus. We recorded in the rat, and Vanya's laboratory recorded in the cat. And if you do that, you find in the medial part of the thalamus, not in the ventrolateral part of the thalamus that's considered to be the important part of the somatosensory system, but in the medial thalamic areas, you find neurons that respond to visceral input and cutaneous input. And we studied colon and esophagus and found that neurons responded to both colonic distension and distension of the esophagus, as well as somatic sensation from the skin and the fur of the animal. So it seems like those inputs from the visceral organs converge on somatosensory neurons before they get to the cortex, and they may be represented or confused at that level. After all, almost all visceral sensation is referred to a cutaneous or other site. And so I don't think you're going to find a selective visceral cortical column, for example, and people certainly have looked. But the imagers out there who look at cortical imaging and subcortical imaging find tremendous overlap within the somatosensory system, and I include the visceral organs within the somatosensory system. They find some areas that seem to be more important for visceral sensation, like the prefrontal cortex, and some areas like that, but not exclusively so.
Thank you very much, Jerry. Maybe afterwards I'll ask something else. But
Yeah,
wonderful,
good talking with you again, Marcello.
Wonderful seminar.
Thank you.
Angela wanted to ask some questions. Angela, your microphone. Your microphone, Angela.
Ahí está.
Perfect.
It was very nice to listen to your lecture. Although I've never worked with the gut, I'm always been interested in, and my first scientific or so-called scientific work was to study the serotonin cells in the gut. So you produce a very nice remembering when you talked about them. So, but the question is related to what Marcelo asked just in his last question, and is, how much do you think of the painful and other sensory information from the viscera is conscious? Or maybe how does it affect what we supposed to be our conscious understanding of what happens to us?
I thought you said you weren't a philosopher.
Well, I said that was before. Now I try to.
It's interesting. I have a doctor of philosophy degree, but I only know about pharmacology and physiology. That's a very difficult question. There's lots of ways to sort of think about that. I mean, among the things that make you feel badly, I think gastrointestinal distress is up near the top of the list. Mm-hmm. Nobody likes to have, be sick and have diarrhea and be nauseous and things like that. And so the gut is really important. And I don't know how to answer your question. Certainly, input from the viscera influences the central nervous system and has some prepotent effects. You can do things experimentally like distend the colon, and then distend the colon while you're crushing the forepaw of the rat or providing another noxious stimulation, and look at interactions. And you can show that there are certainly interactions that are ongoing. But with respect to conscious sensations and things that happen above the neck, I think that I don't have a good response for you. I'm sorry.
Okay. Just, I wanted to know what you thought about it. Okay, thank you very much.
But you know, now that we are on a philosophical ground, I just wonder, as a pain researcher, if you really think that we shall ever solve the problem of pain.
No, pain is always going to be with us. I think the best that we can do, and it's been shown by the kinds of studies that are associated with those people who do cognitive behavioral therapy or mindfulness training, or even yoga and some other things like that, that you can modulate to a certain extent and learn to live with your pain, but I don't think we'll ever overcome pain. We can certainly render people unconscious and they are unpainful, but they're sort of not alive at that point. So pain is going to be with us. I don't think it'll ever go away.
So that takes me to the last question I have, which is how much is known, for somatic pain, we have descending serotoninergic pathways and descending noradrenergic pathways, which modulate noxious afferents, somatic noxious afferents at the level of the spinal cord, descending systems that are in the brain stem. Is there anything similar for visceral afferents?
Yes. It's been shown experimentally that you can activate the periaqueductal area or the ventromedial medullary area, either with a chemical or electrical stimulus, and you can modulate and control visceral afferent input into the spinal cord by recording from dorsal horn neurons that are receiving visceral input. And it's modulated by noradrenergic and serotonergic systems. There's also some thought that there are GABAergic systems and neurons that can contain somatostatin. So it's been studied and some people have looked at that, but it puts us all back in on the same pitch when you're playing football. The players are the same and the results are the same when you try and control them.
Thank you. Thank you so much.
Jorge Aquino.
Thank you, Jerry, for the talk. It was very, I didn't know so much about the subject, so for me it was kind of descriptive and very, I'm happy for that. So I have a question regarding the motility of the gut. It's known that the bacteria sometimes can affect the motility of the gut. So is there any loop, like within the gut, within their nervous system, so that these afferents will modify the function of the neurons in the motility of the gut? Or you may expect a central kind of activity also, like a loop more in the central nervous system?
Yeah, the enteric nervous system of the gut, of the gastrointestinal tract, is really quite amazing. And that's why I was suggested before that we don't know enough about that interaction. But it's been well established. Satish Rao, many years ago, showed that drinking coffee activated a gastro-colon reflex that led to the urge to defecate. And so there certainly are loops within the gastrointestinal tract. And certainly when you start to eat food and it begins to be digested and move into the duodenum, it also initiates a increase in motility that can be recorded down in the colon. So the enteric nervous system was investigated for many, many years by a large number of investigators. Many of them, it seemed, housed or located down in Australia, but we still don't know how the enteric and the extrinsic nervous systems interact. And I think that's an important thing that we need to find out, because I think that the motility of the gut can influence the sensory response to input from the gut. We certainly know that drugs that are used to treat irritable bowel syndrome, like antimotility drugs and serotonergic receptor antagonists, many of them have an effect simply because they either decrease motility or they increase motility and reduce constipation. And it likely due to some interaction that we don't understand well enough at all.
Okay, then also another question is regarding if this afferent can release molecules that can cause inflammation locally in the epithelium, for example, of the gut.
So you mean local gut inflammation?
Yes, caused by these afferents.
Yeah, I'm sure that plays a role because anything that can have access to those epithelial cells can generate release of things from the cells that are from those cells that are close to the afferent endings. We've known that. Lori Berter has been working in the urothelium of the bladder for many years and has clearly established a relationship between urothelium and sensory endings. And so I think local inflammation is important. And it's one of the reasons people have gotten interested in these fecal transplants for people with irritable bowel syndrome, for example. They think that the biomass in the gut is unhealthy, and if they change it from an unhealthy to a healthy status, it's going to improve their irritable bowel syndrome. Well, it turns out that that works, but it's a transient change, and it turns out that doing these fecal transplants is not as helpful as people thought they were going to be.
Yeah, and the last question maybe, it's known that in the lung there are some cells, neurons, that are just below the epithelium of the bronchium. So, and it's known that they have a function in the sensing the lumen of the bronchium. So I don't know if it's very well known, this system, because it seems to be like local, that they just influence in the surrounding, but I don't know if it goes to any kind of ganglion local in there in the lungs, or just as a local circuit.
Yeah, it's a very good question. We did some studies a long time ago looking at irritation of the bronchioles and the lungs by having rats smoke cigarettes. So you could get at that time from the National Institute of Drug Abuse cigarettes with a known amount of nicotine. And so we connected that to a respirator, and we were recording from neurons in the thoracic spinal cord, and we're able to show that ammonia and smoke and other stimuli activated those neurons. There's an investigator who's primarily studying asthma at Johns Hopkins University, and his name escapes me at present, but he's recording from afferents that innervate the lungs and the bronchioles. And he has gotten involved in the pain field because he's found a number of these neurons that respond to these noxious stimuli. And it's an understudied area. And if you're not doing things on weekends, you could get involved in the lab and start to study afferents innervating the lungs. What do you think of that?
Well, actually, I was looking at some mice that are reporter mice, and we could find these neurons just below the epithelium. So it was like something that amazed us a lot. Yeah.
We think we know a lot, but if you look at back in time, how far have we come from what James Mackenzie was thinking and talking about more than 100 years ago? And who knows what people are going to be talking about 100 years from now. We don't know very much about many things, to be honest with you. We think we're doing great work, and it's important that we do that work, but we're still pretty ignorant when it comes down to it.
There's still a lot to do, so it's good for us.
Well, thank you.
Thank
you for letting me into your homes. When you do a Zoom talk, you get to see where people live. It's
Yeah.
different than giving a talk in a lecture hall. So thank you for letting you into your homes. I'm sitting in our bedroom. As you can tell, there's some pillows over my shoulder, and there's a door here that goes out to a terrace. My wife is in the other bedroom working on her computer. So this is how we live out in Colorado.
It looks nice. It looks very nice.
Thank you very much.
you.
I don't know if there are any more questions. Anyone would like to place one last question, perhaps? Seems that everyone is satisfied. I will call this meeting to an end so that everyone can rest after such an intense lecture and question sessions. Thank you so much, Jerry. It was wonderful to hear you, to see you again, and well, to wish you well, to continue to continue so that everything continues well over there in Colorado. In the lovely picture that you're showing is kind of representing that.
So as you can imagine, my screensaver is a series of pictures of the mountains.
Yeah, just as it was in your office. I remember that very clearly.
Well, thank you all for your attention. I very much appreciate it. It was great seeing you again, Pablo and Carly. Good to see you as well. I hope things are going well. Bye-bye, Marcelo.
Thank you so much.
Bye-bye.
Bye-bye.
Bye, Jerry.
Thank
Bye-bye.
you, June, Dean,
Bye,
everyone.
Pablo. Bye, Dean.